Brad Williams, Excel Prime.
Analysis of variance, or ANOVA. The ANOVA analysis tool provides different types of variance analysis. The tool that you should use depends on the number of factors and the number of samples that you have from the population you want to test. We have a single factor ANOVA. This tool performs a simple analysis of variance on data for 2 or more samples. The analysis provides a test of the hypotheses that each sample is drawn from the same underlying probability distribution against the alternative hypotheses. That underlying probability distributions are not the same for all samples. If there are only 2 samples, you can use the worksheet function t-test. With more than 2 samples, there's no convenient generalization t-test, and the single factor ANOVA model can be called upon instead. ANOVA 2-factor, the analysis tool, is useful when data can be classified along 2 different dimensions. For example, in an experiment to measure the height of plants, the plants may be given different brands of fertilizer, for example, A, B, or C. It might also be kept at different temperatures, for example, low and high. For each of the 6 possible pairs of fertilizer and temperature, we have an equal number of observations of plant height. Using the ANOVA tool, we can test whether the height of a plant for the different fertilizer brands are drawn from the same underlying population. Temperatures are ignored for this analysis. Whether the heights of plants for the different temperature levels are drawn from the same underlying population, fertilizer brands are ignored for this analysis. And there's also a 2-factor without replication. The analysis tool is useful when data is classified on 2 different dimensions on the 2-factor case with replication. However, for this tool, it is assumed that there's only a single observation for each pair, for example, fertilizer temperature, in the preceding example. Let's just use a simple single factor in this example. Here we just have low, medium, high, and control, and this is the output. Very straightforward, very simple. What we're going to do in order to grab this information, we're going to go up to Data Analysis, go to Analysis, ANOVA single factor, hit OK. And then here's the dialog box. It's very straightforward. All we need to do is put in some data. So we have an input range. We have A2 to D8. And make sure you're within the actual box. We're grouping by columns, labels in first row, alpha. You can change this. That's just your confidence level. Your output range, we want it over here in O. You can put it anywhere. You put it on a new workbook or a new worksheet. We're going to go output range, hit O, and hit OK. The range is because we're messing around with too many things. This is very sensitive here. This input range, make sure you're in it. Our output range, if you're going to make changes, don't click a cell. If you're already in this box here, try to click out of it. This thing is tricky. Just be patient here, because if you're anywhere outside of this box and you click any cell, it's going to disrupt this input. We have an output cell. If you have an output, just make sure you're in the box, click this cell and hit OK. There we have a simple factor. Now it's going to be different. It reads a little bit different. You can see are different. Go figure. Why? Because the labeling is really screwy with this tool. So you go delete, and to get rid of all this stuff, just highlight it, hit delete, go back in to data analysis, go to single factor, go labels in first row, A2, A1, and see, this thing is impossible. It's typical Excel. Sometimes you just have to keep fussing with it in order to get it to distribute what you need. Go here, go to data analysis, go to OK. Now we're going to change the range. We're going to capture everything. We're going to say labels in first row. Our output's the same place. Hit OK. And see, this time you have to explicitly place the labels in the first row. It's not as though you're inserting a table. Here it's a lot different. You actually have to highlight the entire range, and then if there are labels or headers, you have to state it explicitly. There's your output. So we have low, medium, we have a range of 7, we have a sum, we have an average, a variance. If If you don't know your degrees of freedom and the other factors, break out your statistics book and review what this information means. But that's just a quick introduction to the ANOVA single factor.
Brad Williams, XL Prime.